Right, hi everyone. Let me just share my screen. Uh, can everyone see my screen? All right, perfect. All right, my name is Sam, and for the first presentation today, I'll be going over the structure of DNA. All right. So for this presentation, I'll be going over three main aspects of the DNA double helix that was covered in lecture. First, I'll be going over how nucleotides are brought together in DNA and RNA, and more specifically through the sugar phosphate backbone. So as you can see, there's a double strand, and each double strand is made of sugars and phosphates. Then I'll be going over the base pairing between these two strands, which is done through the nitrogenous bases, adenine, thymine, guanine, and cytosine. And I'll be talking about how through these base pairing, it forms the DNA structure and it also provides stability. And then finally, I'll be talking about the major and the minor grooves, and I'm talking about how understanding these are very important to know how DNA and proteins interact. So with that being said, let's get started with the sugar phosphate backbone. As you can see here, DNA has a 5' end and a 3' end. The 5' end comes because when naming a sugar, there's 5 carbons. It starts with the 1', 2', 3', 4', the 5'. At the 5' end, there's always going to be a negatively charged phosphate group. And then on the other side, there's a 3' end, which there's going to be a hydroxyl group. And when this sugar phosphate backbone is forming, it goes from the 5' to the 3' end. And this happens because the very last 3' hydroxyl group will attack the alpha phosphate of a nucleotide. As you can see in this picture, the hydroxyl group is the nucleophile, and it attacks the alpha phosphate, forming a phosphodiester linkage. And this interaction is favorable because, as you can see here, this first phosphate group and the second phosphate group have negative charges very close to each other, and this isn't favorable. So by forming this and by cleaving off this pyrophosphate, you're relieving that repulsion between the two negative charges. And two other things that I want to point out in this sugar phosphate backbone is the fact that because this is DNA, the sugar is a deoxyribose. And I underline deoxy because that means at the 2' carbon, so meaning this carbon here, there is no OH group. And this has, but in RNA, it's a ribose, so meaning there is an OH group here as well. And this has very important implications in the overall structure of DNA. And as the other tutors will get into later on in this concept review session, the fact that there is no OH group here makes it so that DNA can adopt a B form or a C2 endo conformation. And this will lead to it having wider major grooves, which I'll talk more about later on. And also, not having this 2' OH group is also favorable because this 2' OH group can also attack and actually self-cleave or break off the sugar phosphate backbone. So an RNA which has this OH group is less stable because it has this self-cleaving aspect as well. And then moving on from the ribose, there is these phosphate groups, which are, again, negatively charged. And this also has many important implications in the structure because since they're negatively charged, the DNA structure makes it so that they're as far away as possible from each other. And also it can form interactions with polar groups that surround the DNA, like water, for example. And if there's no questions, I'll move on to the inside of the DNA, which is the base pairing. So you can imagine these two strands as being the sugar phosphate backbone that I just talked about, and inside there's the nitrogenous bases. And if you want to connect what I just talked about to this, the connection is here, which is the glycosidic bond, which connects these bases to that deoxyribose, which I was just talking about. And as for the bases, there's two groups, purines and pyrimidines. Purines are adenine and guanine, as you can see here and here, which are two cyclic groups, and then there's the pyrimidines, which are thymine and cytosine. And it's always the interactions are always going to be between one purine and one pyrimidine. So it's always going to be adenine, thymine, and then guanine and cytosine. And this, an analogy that made a lot of sense to me last semester when I took this class was, it's almost like a zippering effect. When one purine and pyrimidine bind together, it's almost like a zipper that it kind of leads to more and more base pairing happening. And this base pairing is referred to as Watson-Crick base pairing. And in DNA, it's always followed, but there's some exceptions that I think other peers will talk about, that there's some exceptions to this base pairing not happening. But for the most part, it's going to be Watson-Crick base pairing. And then this is just a quick question. Based on this picture, would you think that GC bonds are stronger or adenine thymine bonds? And people can type in the chat, or if anyone wants to unmute and say it out loud.
I would assume it's G and C.
Yeah, exactly. Because as you can see in the picture, it's forming 3 hydrogen bonds, and adenine and thymine are only forming 2 hydrogen bonds. So generally, DNA with a higher GC count has stronger base pairing between the 2 strands. Good job. And now that we talked about the 2 aspects, we can bring it all together. So DNA runs in 2 anti-parallel strands, and that following based on what I said from the first slide, that one strand has the 5' group at the top, and then it runs down to the 3' end. And then the other strand has the 3' end with the hydroxyl group and runs down to the 5' end with the phosphate group. And then there's the sugar phosphate backbone on the outside, and then there's a base pairing with the nucleotides on the inside. And one other thing that I want to talk about is the handedness of DNA. Other tutors will definitely get into the 3 major forms of DNA, but for now, I just want to introduce you to the major form of DNA, which is going to be right-handed. And I know this is kind of a confusing topic when I took the class last semester of seeing how it's right-handed or left-handed. But if you, the way I learned, the way that worked out for me the best was I did the using my fist, using my hand. So if you can wrap DNA with your right hand, it's going to be right-handed. But if you can't, and as you can see with this left picture, you can only kind of wrap it around with your left hand with your thumb pointing up. So it's going to be left-handed. And another thing that worked for me, if this doesn't work for you, is if you can look at these ribbons or these strands, I guess, that are towards the front. In this case, it's pointing towards the right, so it'll be right-handed. And in this case, it'll be pointing towards the left, so it'll be left-handed. And so that's another thing that worked out for me. And this is a very important concept throughout this course, because as you'll later see, proteins as well, the alpha helix can also be right-handed or left-handed. So knowing this, making sure you clearly know what is a right-handed and what is left-handed is very important. And then if there's no more questions, I can move on to the final concept I'll be going over, which are the major and the minor grooves. And these arise because when you're forming the double helix, some grooves are going to be larger than the other ones. And this arises from the fact that DNA adopts either B form, A form, or Z form. And the tutors will get into this more, but the major groove is the groove that's wider and is more accessible, is in the B form, and the minor groove is the one that's narrower and less accessible. And this has very important implications when it comes to proteins binding to DNA. And there's many processes in the body that require this interaction. For example, DNA synthesis. In DNA synthesis, proteins have to come and read the sequence, or in RNA synthesis, again, proteins have to come and bind to the DNA and read the specific base sequence of the DNA. So having these major groups that are accessible is very important. So because you want these alpha helices and beta sheets to come in and interact with bases inside the DNA. And if you zoom in into each of these parts, you can see a picture like this, where the major groove is the one that's convex, and the minor groove is a smaller one that's concave on the bottom. And there's always going to be a sequence-specific recognition of DNA where there's specific polar and non-polar groups sticking out. And just as a last exercise, if anyone wants to unmute and identify each of the groups as either hydrogen bond donors or acceptors or just an hydrogen atom. Let's say for this, the major group. So from left to right, if you can identify what each group is.
It would be from the left, hydrogen bond acceptor, and donor. And the last one, it's not a donor or an acceptor. It's just like a H atom there.
Yeah, perfect. Good job. That was right. And this is very important because for guanine and cytosine, it'll be different from adenine and thymine. And that's why it's going to be sequence specific. And yeah, that's it. If no one has any questions, that's my part today. All right.
Hello, everyone. Can everyone hear me clearly? Yes? All right, okay.
Yeah, we can hear you.
All right, let me share my screen. Hello, everyone. I am Yishen. You can call me Lisa. I am a new undergrad tutor. I took class last semester, and I'm very happy to work with you all this semester. So let's talk about the DNA double helix stability today. I already posted the blank handout in the Google Drive. Yeah, I invite you to fill in the notes with me just to keep yourself engaged with the session, but I will always post a refilled version afterwards. So for this concept, one point I really want to emphasize is the formation of DNA double helix is always in an equilibrium process. Or we can also say it is in a dynamic process. That means although we'll have this extreme prevalency of observing DNA in its double-stranded conformation, there is always a non-zero chance to observe a DNA not in its double-stranded conformation. So we want to think about the force underlying those two conformations. So from the bird's eye view, let's fill in the blanks together. For the force varying the double helix formation, we know there is hydrogen bonding. There's base stacking, which consists of electrostatic interaction. And also van der Waals attractive force. And there's also metal ion interaction. And for the force opposing the double helix formation, we know there is competition for hydrogen bond from water. And there's also very important entropy consideration upon the formation of double helix. So let's talk about hydrogen bonding first. We talk about hydrogen bonding in the context of base pairing, of which we know one characteristic is cooperativity, which means the formation of first base pair via hydrogen bonding will favor the subsequent formation of base pair. And that means there's a lowering energy barrier of subsequent hydrogen bonding. But then we also know hydrogen bonding is highly affectable by its environment. Here we mean water. So the competition from water will significantly weaken the energetic gain from hydrogen bonding. That means in a double-stranded DNA, nucleotide base will hydrogen bond with another base following Watson-Crick base pairing rule. But the base can also hydrogen bond with water molecules if it's in a single-stranded state. And as long as the hydrogen bonding requirements are all fulfilled, the energetic difference between two cases is actually very small. As you will recall, the effective strength of a base-base hydrogen bond is comparable to thermal energy. And that means at room temperature, if you just look at one base pair, the hydrogen bond between them is fairly easy to break. So I have some questions that you can look at later on, maybe if we have time. So although hydrogen bond is not a major contributor to the energetic favorability, it does highly related to the accuracy or fidelity in DNA replication process. And for a complete understanding of that topic, I recommend you to read chapter 19 after sufficient preparatory work. But today we can think of this problem by asking ourselves, what if there is a mismatch in hydrogen bond? So as you can see, if there's a mismatch in hydrogen bond, that means there's unfulfilled hydrogen bonding requirement. So those pairs, right? And maybe the unfulfilled hydrogen bond introduce a large energy penalty that enforce the base to behave in a standard pairing way. But another very important or more important factor to consider is the shape. As you will recall, in standard base pairing, each purine pyrimidine set has uniform shape, whereas in a mismatch base pair, you can see there's a clearly asymmetrical structure that can be easily detected by DNA polymerase. So the consequence of that is either the wrong nucleotide cannot be incorporated into the new strand, or it will be more likely to be removed in a proofreading process. And that contributes to the fidelity of DNA replication. Okay, so let's talk about base stacking, which is the driving force for DNA double helix formation. Why there is base stacking? So if we look at those base, we see these purine pyrimidine, they are highly polarized substituted aromatic rings. And they have heteroatoms like oxygen, nitrogen. They create large dipole moment with carbon. And because of these, we have this significant electric polarization in each base. And base stacking is the solution DNA offers to mediate those electric polarization. So from a molecular level, two adjacent bases in one strand, they will rotate against each other to achieve the favorable overlapping of partial charges. So the main intramolecular force involved in this process, one is very obvious electrostatic interaction. Yeah, I'll post a nicer version later on. So notice the rotation, it is the rotation that gives the helical structure. And also notice the rotation is roughly about 36 degrees. And it is also non-sequence specific, which means that whatever, it doesn't matter which base is stacking upon. And later on, you will know that the distortion runs 36 degrees. That gives rise to the supercoiling phenomenon because it introduced torsional strain. Sam mentioned GC-rich sequence is more stable than AT-rich sequence. And I'm here also suggesting that you can think of the theoretical overlapping of partial charges. They also have the contribution to this stability. The second intermolecular force that is involved is van der Waals attractive force or van der Waals interaction. Because atoms are brought into close contact. More specifically, they're in van der Waals contact. And this can be testified by the simple observation that rise per turn is roughly 3.4 angstrom. If you're referring to the van der Waals radii table, right, you can easily reach that conclusion. But something I want to mention is we know van der Waals interaction is weak, but the combined effect is very significant. If you remember the Geico example from yesterday Tutoring session. So the planar and flat aromatic ring, they maximize the possible van der Waals contact by offering this large interacting surface. And this is a practice question that if we can go over if we have time, but it should be clear. I offer a lot of hints. So there's another thing, metal-ion interaction we should consider because we know there is significant charge in the phosphate sugar backbone. So the metal-ion, metal-cation, they help show the electrostatic repulsion between phosphate group. Notice metal-ion usually exist in hydrated state. For example, magnesium 2+ coordinate with 6 water ligands. There are 3 types of interaction. They are categorized based on how directly metal ions is interacting with the nucleic acid. You can fill in by yourself. They're pretty self-explanatory just based on the figure. Notice this one is most common. And notice metal-ion interaction, they allows the tight packing of DNA without electrostatic repulsion being a large issue. Then maybe you can look at this figure and tell me which type of interaction it involves in the chat or later. It's really clear. The last thing I want to talk about is entropy consideration. Entropy is a topic you will learn extensively later on. But as for now, we can think of entropy as the freedom to adapt different conformation. So the more conformation a structure is able to adapt means it is in a state of higher entropy. And we know nature loves to maximize entropy. So for double-stranded DNA, there is usually just one stable conformation. But for single-stranded DNA, we can actually calculate the number of conformation it can adapt based on a set of parameters such as the backbone angle, the conformation of sugar, the base orientation, those calculations you'll go through later on. But without even in calculation, we can be sure that double-stranded DNA is in a lower energies, at least in a lower entropy state than single-stranded DNA. And this decrease in entropy is a major reason why DNA double helix will not be elongated infinitely, despite all the favorable energetic interaction. Okay, some takeaways, very quick. Hydrogen bonding, base stacking, metal ion, they altogether shape the structure and functionality of DNA double helix. Some structure elements you can think about, such as the uniform shape of base pair given by the canonical hydrogen bonding, the helical structure with 36-degree twist given by the favorable overlapping of electrostatic interaction, favorable overlapping of the partial charge region. And that gives rise to 10 base pair per turn, roughly. And because atoms are brought into close contact, rise per turn is roughly 3.4 angstrom. And also, Sam mentioned the phosphate group will try to be separated from each other as far as possible, whereas we also have this base stacking that tend to bring atoms into close contact. So those are two opposite force. Some function you can think about, one very primarily I can think of is the fidelity of DNA, the fidelity in DNA replication process. Yeah, so I think you guys can look over these questions by yourself, because yeah, I'll let other tutor go through their presentation. Is there any questions? No? All right, thank you.
Okay, hi guys. Thank you for coming to our first Thursday tutor session. Can everyone see my screen? Sorry, I can't see your videos, so someone will have to tell me.
Yep.
Okay, thank you. Okay, so basically Sam already started kind of talking about B-form DNA. I'm going to be going more in depth into the three kinds of helices that we cover in this class, A, B, and Z. So first of all, this course basically, I would say, covers two major ways to describe the different conformations of DNA and RNA. And the first has to do with the orientation of the base relative to the sugar. And so first, let's define what the bond is called, the bond that's between the base and the sugar. So this is our glycosidic bond. More specifically, this is the bond between the C1 prime of sugar and the N1 or N9 position of your pyrimidine or purine base, respectively. And because this glycosidic bond is a single bond, it can rotate, but that doesn't mean that it can just rotate to give an infinite number of conformations because of steric clashes. And so upon rotating, different sterics give rise to the two most common or to the two most lowest energy conformations, which are termed anti and syn. So the anti is when the sugar H1 prime and the base C6, if it's pyrimidine, or C8 position, if it's purine, are trans to each other. And the syn is when the H1 prime and C6 or C8 are cis to each other. So I've just highlighted in blue the most important bonds that I think shows more easily the trans conformation in the anti and the cis conformation in the syn. So the second way that we sort of describe the conformations of DNA and RNA is what is called the sugar pucker. So as you guys talked about in lecture, the sugar ring cannot be planar, because if it was, there would be too much steric clash between the different substituents of the sugar. So instead, what it does is it adopts something called a sugar pucker, where one atom of the ring sticks out of the plane while the rest are coplanar, meaning that they are in the same plane. So a sugar pucker can be either endo or exo. Endo is when the out of plane atom is on the same side of the plane as the C5 prime position, whereas exo is when the out of plane atom is on the opposite side of the plane from the C5 prime. So if you were to have a C2 prime endo sugar pucker, that would basically mean that all of the atoms in the sugar ring except C2 prime are within the same plane. But the C2 prime is out of that plane, and it's also on the same side of the plane as C5 prime. And with C3 prime endo, it's pretty much the same thing, except just with C3 prime instead of C2 prime. So there is an important difference in the sugar pucker between DNA and RNA. As Sam mentioned, the sugars in DNA and RNA obviously have very different properties because of the 2' hydroxyl group that RNA has, but DNA doesn't have. So basically what this entails is that DNA can be in either the C2' endo or C2' endo if it's B form, or C3' endo if it's A form, whereas RNA can only be C3' endo. So why is this? This is because for a sugar to be C2' endo, its 2' substituent and 3' phosphate group have to be very close to each other. And so if you have a DNA molecule, then its 2' substituent would be a hydrogen atom, and hydrogen atom is really small. And so it's small enough so that it can be close to the 3' phosphate group without serious repulsion. And so C2' endo is allowed in DNA. Whereas in RNA, its 2' substituent is a hydroxyl group, which is significantly larger than a hydrogen atom. And so being that close to a phosphate group is really not favorable. And so that conformation is not allowed. So if you take away anything from the slide, it's that RNA cannot adopt C2' endo and therefore cannot form a B form helix. So what are some properties of B form DNA? As we mentioned before, it has a C2' endo sugar pucker, and it is in the anti orientation, and it's also right-handed. The helix is right-handed, and the base pairs are completely perpendicular to the axis of the helix. So axis of helix meaning a line, like if you were to run a line through the helix, and the base pairs would be like horizontal to that, and they would form a 90-degree angle. And so they are perpendicular to each other. And also each base pair is centered on the axis. And also the helix forms 2 grooves of unequal size, as we talked about. There's the major groove, which is wider and more accessible and has more information content, and there's the minor groove, which is narrower and less accessible, and also has less information content. What do I mean by information content? So going back to the idea of protein proteins interacting with DNA, obviously proteins like, for example, transcription factors have to be able to read the DNA sequence, and they actually do this without opening the helix by reading the outside of the DNA via the major groove. And so how they do this is they recognize patterns of interaction sites that are specific to each of the 4 possible base pairs, which are GC, CG, AT, and TA. So what this means is that each of those 4 possible base pairs has a specific and distinguishable pattern from each other that is made up of the 4 types of interaction sites, which are, as we covered, the hydrogen bond acceptor, hydrogen bond donor, hydrogen atom, and hydrophobic group, which is usually a methyl group. So you might be asking, why can't proteins read the minor groove? Well, that's because the patterns of the G, C, and CG, as well as the AT and TA, look exactly the same. So this is more clear if you look at this picture. So if this is a GC base pair and this is a CG base pair, if you look at the major groove of the GC, it's different from the CG. But if you look at the minor groove, the patterns are the same. They're both red, blue, red, red, blue, red. So if you're a protein and you're trying to read this, read a DNA sequence, but you're in the minor groove, you wouldn't know, is this a GC or a CG? So it has less information content than major groove. And then, so there's the second type of helix, which is A form. As we talked about, the A form has a C3 prime endo sugar pucker. It's also in the anti-orientation, and it's also right-handed. But unlike the B form, the base pairs are not perpendicular to the helix axis, so they're a little slanted, and they're not centered on the helix axis. And what this results in is that the A form major groove is narrower and deeper than the B form major groove, and also the minor groove is wider and shallower than B form minor groove. So major and minor grooves are still present in A form, but compared to B form, the major groove is less accessible to proteins. And then lastly, I'm going to cover Z form DNA. So this kind of DNA has a sugar pucker of both C2 prime endo and 3 prime endo. It alternates between the two. And what that means is that only DNA can be Z form and not RNA because of the C2 prime endo sugars. And it's also switches between anti and syn. So there's the T and C nucleotides are anti, whereas the A and G nucleotides are syn. And it's left-handed instead of being right-handed. So what an important thing to know, important consequence of that is that if a region of B DNA switches to Z DNA, the segments of DNA at the junctions between, the different helices, so circled in blue here, have to melt in order to accommodate the left-handed Z-form helix. And Z-form is formed in GC-rich stretches of DNA. So like so it'll probably be seen in sequences that are like GC, GC-rich. So yep, that is end of my slides. If anybody has any questions, you can unmute yourself or post it in the chat. But that's the end of my slides.
Okay, so then I will share my slides.
Oh, I'm so sorry. I just realized somebody asked in the chat, could you go back a slide? I didn't see that. Do you know what slide that was?
It's okay. I can just check later on the Google Drive.
Okay, great. Sorry about that.
That's okay.
Okay. All right. Hello, everyone. I'm Poojan, and today I'm going to talk a little bit about DNA supercoiling. So this is something that you touched upon in, I think, yesterday's lecture. So the first thing I want to start off with is a question of what exactly is supercoiling? So here is sort of the kind of figure representation of what it would look like. And as you can see, there's like a circular piece of DNA like this. And as a result of supercoiling, it ends up just kind of bending and twisting on itself. So supercoiling is not just when a circular piece of DNA or any piece of DNA just happens to fold on itself, but it's actually because there's strain imposed on the DNA molecule as a whole that forces it to adopt this conformation. And if you were to actually observe this happening in vivo inside of a prokaryote like a bacteria, this is typically what it would look like. And when we talk about supercoiling, there is also one important parameter that we use to quantify the level or the extent of supercoiling, and that's this notion of ride. So ride can be positive or negative depending on the way that the supercoiling occurs, which I will discuss a little bit later. But the main thing to know for right now is that basically the number of crossings that occurs with a supercoiled DNA molecule, that indicates the ride. So in this case, there are 2 crossings right here, and here there are 1, 2, 3, 4. And then if we knew whether they were left-handed or right-handed, we could say that this is -4 or +4. And just as one thing I want to point out, when I write T-mol, that just indicates the molecules of life. That's your textbook. So if you want to look at this figure, it's figure 2.3 in your book. Most of these figures that I use either come from your book or just quick Google searches. And so now that we kind of have an idea of what supercoiling is, or at least what it looks like, the next thing I think that kind of is a natural question is, what exactly causes supercoiling? And so to understand this, we have to step back a little bit and think about the structure of DNA, which I think the tutors today have done a really good job of explaining. So if we recall, base stacking is one of the most important driving factors in terms of not only stabilizing the DNA helix, but forcing it to adopt a certain conformation. And here, just to illustrate kind of what makes base stacking so powerful, you have 2 nucleotides, cytosine and guanine. And as a result of the way that their electron clouds are just in particular configured, the overlap is such that if the cytosine is offset by about 35 degrees or 36 degrees, that's when you get a really favorable overlap like this. And so this is why when you have more and more nucleotides in a growing DNA helix, they're all offset by about 36 degrees. And the main point that I want to make through this slide is that this 36-degree offset is pretty important, because if you were to try to deviate from that optimum, notice that there are these 2 regions that are both very electropositive. So if you instead try to rotate this space a little bit more in the clockwise direction, you would end up with 2 electron poor regions overlapping, and that would make this not so stable anymore. So really what happens is that the 36 degrees that's between every single DNA base, that's really enforced at the level of base stacking. And so what that means for the energy of the DNA molecule as a whole is that if you subject DNA to like torsional strain, meaning that you take a long strand of circular DNA and you twist it, it's not really a good option for the DNA to kind of dissipate that strain by changing the angles in between all of its spaces. And it's more energetically favorable to just dissipate that through globally having the entire DNA molecule bend on itself slightly. And that's what causes supercoiling, because it's more energetically efficient, or it requires less energy to dissipate strain through a global deformation than it is to just change the angle in this manner. And so with that being said, I kind of want to give you some intuition for what right-handed and left-handed mean in the context of supercoiling, because this is a pretty important concept and it comes up a lot. And I think we've had a little bit of a discussion of what this intuition would look like. But in this case, here on this picture on the left, this is an example of a right-handed DNA helix. And here you have an example of both right-handed as well as left-handed. Oh, there's a question in the chat. Okay. Well, let me go back to the last slide. Yeah, so did you have a question about the angles in particular?
Yeah, so my question was just, do these, I guess, sticks or whatever you want to call them, do they each represent like a base pairing?
The sticks? Oh,
The
these
upper right, yeah.
Oh, yes. Yeah, so like these purple and orange dots are like the sugar. And yeah, you can think of the bar as like I guess the halfway point would be like, this is like one nucleotide and this is another nucleotide. So this entire thing as a whole is like one base pair.
Okay, and then this is like what makes the structure, like the mix in structure, right? Like the angles, like I guess the double helix, or no?
Yeah, I'm not sure if like, oh yeah, go ahead.
From this like way of alternating.
Yeah.
Like the 36 degree rotation, like is that why we have the double helix, or like why it rotates like it does?
Yeah, that's exactly why. Yeah.
Thank you. Sorry, that was a little weirdly.
Yeah, no, that's fine. That's fine. Yeah, I highly encourage questions. Yeah, and so now just to go on to this intuition of like what makes a DNA molecule left-handed versus right-handed, here you see an example of a right-handed helix. And one way that I like to think about this is that if you were to pretend that this was a stairwell and that this red line was like the handrail for the stairwell, you could think about which hand would you use to grip the stairwell as you're going up. And in this case, it would be your right hand as you're ascending the helix. And then in this other case, you can kind of imagine having like a cylindrical container like this with a ribbon wrapped around it. And here in this case, you could ask yourself, if I want to take my palm and make sure that it's always in contact with the ribbon, how would I maneuver my hand around the cylinder to make sure that I'm always gripping the ribbon? And would I use my left hand or my right hand? And in this case, as you can see, your palm would have to face the inside of the ribbon, and your palm would have to face the ribbon that's on this side of the container. And as your hand would have to go around the back to continue making contact with the ribbon, that's why you'd have to use your right hand in this case. Whereas with this case, you'd have to use your left hand in order to make it all the way around the cylinder. And that's kind of where the intuition for left versus right comes from. And don't worry if this doesn't make like complete sense right now. After you see more examples, I think you'll be more comfortable with it. So in this class, there are two main ways that you will see DNA supercoiling manifest. So one is the plectonemic form, which to me looks like a figure 8. And so in this example of supercoiling, the question that I would like to pose to you is this an example of a left-handed crossing or a right-handed crossing? So just try to think about the intuition from the last slide and just feel free to just type in the chat whether you think this is right-handed or left-handed. Okay, I'm seeing a couple of votes for left-handed. Yeah, so this is an example of left-handed crossing, so that's correct. And the reason for this is that if you look at the way that this green strand is coming in front of the yellow strand, you can imagine trying to take your left hand, and as you follow the green strand, as soon as you get to this point, you will need to curve inwards, and only the fingers on your left hand would be able to do that. Because if you had instead started with your right hand here, as soon as you get to this point, you'd have to bend your fingers this way, and that doesn't work. So this is an example of a left-handed crossing. And here's an example of what a right-handed crossing will look like. And so just to verify, you can try to take your right hand, and once you get to this part over here, you need your right hand to make that curve. And so next, what type of crossing is this? Is this left-handed or is this right-handed? And then what is the overall ride? Feel free to just add it into the chat. Okay, I'm seeing many votes for left. So yes, this is also an example of left-handed crossing. And because there are two instances of this, it's +2 for supercoiling, because here, left-handed in plectonemic form is positive supercoiling, so W is +2. And so another way that you will see supercoiling manifest in this class is solenoid form. And this is what it typically looks like. Whenever I see DNA in this form, I think for me personally, it just helps to rotate it counterclockwise, sorry, so that it looks like this. And as you can see here, if we use the same kind of intuition from the previous slides, this is also an example of a left-handed crossing. And here there's the slightly counterintuitive fact, which is that although this is a left-handed crossing, the ride is actually -2. And this comes back to the definition of how positive versus negative supercoiling is defined, which I will just explain very quickly in this slide. So the main point I want to make here is that if you start off with a plectonemic form that has, for example, a right-handed cross, and if you just manipulate it very simply, you can get to a solenoid form that has now a left-handed cross. So even though the supercoiling does not itself change, just by converting from plectonemic to solenoid, you change the handedness of the crossings. So here I have left-handed crossings, but as soon as I convert it to solenoid form, now the crossings are right-handed. So nothing really changed. You just manipulate the molecule as a whole, and these two are considered topologically equivalent. And the reason why this is positive supercoiling is because the convention is defined in terms of the solenoid form. So because you have a right-handed cross in the solenoid form, that's considered positive supercoiling. But of course, when you go from solenoid to plectonemic, the handedness of the cross will change. So now you end up with a left-handed cross, and then becomes, it's still positive supercoiling, but that's why left-handed in this case would indicate positive, whereas left-handed in solenoid form would indicate negative. I know that's a little bit confusing, but I hope that these slides will serve as a reference in the future if you ever need to refer back to this. And the last thing I want to touch upon is, what determines the type of supercoiling that we're going to get? Because we've discussed like left-handed, right-handed, positive, negative. And this is something that you'll probably discuss in lecture tomorrow, but there's one really key equation that is very relevant for this course, which is that linking number is equal to Reith plus twist. We already talked about Reith a little bit, and twist essentially just refers to the number of times the DNA helix makes a full rotation. So because there's a 36-degree rotation between each base, every time you have 10 bases, you make one full rotation. That counts as like one twist. And so whenever you have a situation in which your DNA is clamped between two proteins, then if you increase the twist, you need to decrease the Reith, because if you clamp the DNA between two proteins, the linking number is fixed. And that's typically what determines whether you will get positive or negative supercoiling. And I think you'll discuss that more in lecture tomorrow. So I believe I'm out of time. So anyone has any questions, I can try to answer them very quickly before the next session starts. Okay, great. So with that, I'm going to stop the recording.